In 2.76, we're going to introduce something called DNA transcription. So the central dogma of genetics, or the big idea, if you want to think of it that way, is that a gene equals a protein. And I think I've said this a couple times, but our genes, right, are on our DNA. And the problem with that is that where the proteins are made, if gene equals protein, the proteins are made out here on something called ribosomes, right? Our ribosomes are out here, and that's where protein synthesis happens. And if you can see down here, this is supposed to be a ribosome, way bigger than it really is, but just so you can see. But the DNA is trapped inside of our nucleus. So we need to create a messenger to take that message of what the segment of DNA that we want to express, take that message and bring it to a ribosome, because you can make that protein. And that gene can be many things. That gene can be a hormone, so maybe it's insulin. It could be a pigment that will color your eyes blue or your hair brown. So that's what we mean by gene equaling protein. And transcription is the first process to get there. Later on, we will look at this other process called translation. But first, we need to get the message out, and that message is carried on a molecule called RNA, specifically messenger RNA. So let's talk about the steps. Very simple diagram to explain it here, and maybe a couple confusing things. So when we're talking about transcription, we're going to be talking about this formation of messenger RNA. There's different kinds of RNA, but messenger RNA is the one that has the code for the gene, and that messenger RNA is complementary. Remember, we talked about complementary base pairs in DNA. So it's complementary to the DNA that it's copying from. So I am going to redraw our DNA over here to explain what that means. And also, the last thing we need to talk about is this enzyme called RNA polymerase, which is nice because there's only one that we need to know. So I'm going to put some nucleotides here. So our bases are A, T, C, I'll put another C, A, and maybe a G, and another G down here. So what happens is that this enzyme called RNA polymerase, which they have as this blue molecule down here, and normally they draw it a little bigger, it will separate our strand of DNA. Now, if you remember, the enzyme that did this in replication was called helicase, but the enzyme that does that during transcription is called RNA polymerase. So it will separate that strand, and now we have our bases exposed, right? So these bases are exposed on this template, the one we're copying from. And something that is very confusing is a strand that we copy from on the DNA is called the antisense strand. This one does not get copied for making messenger RNA. So we have our DNA exposed, and then nucleotides will connect. So we have all these RNA nucleotides. We have A's, we have T, well, no, not T's. We have C's, we have G's, and we have not T's. If you remember, RNA does not have the base thymine. Instead, it has something called uracil. And then these free nucleotides will start forming or start connecting. So we have our U, we have our A, will complementary base pair there. C and G don't change. A and U for RNA. We have a C and a C, a cytosine and a cytosine. And I'm going to draw the backbone here. So those nucleotides connect to those exposed pairs. And then the RNA polymerase, just like DNA polymerase, not only does it separate, but then it will connect those 5' and 3' carbons, right? It will connect that phosphate to carbon number 3. So we'll get a solid backbone. And then our messenger RNA is free to leave the nucleus and go find a ribosome. So that's how transcription works. So we're going to form messenger RNA. That is my green piece here. It will complementary base pair to the antisense strand of DNA, and it will be made or synthesized through the help of this enzyme called RNA polymerase that has two jobs. It separates and it binds those carbon number 3s to the phosphates along our backbone there of an RNA.